Can we start the session? So I think, Pooja, how many
Sir,
participants?
sir, wait for 2 minutes. I think the live is not yet started. 15 participants are there, sir. Sir, you can start now, sir.
Okay, how many participants now?
15.
Okay. So we are going to start this session. That is tutorial 7. Uh, tutorial 7 with me, Orindita, PhD scholar of our department. So tutorial on the hands-on demonstration, some short-based learning techniques, and basically in unsupervised method, Orindita will assist me and also demonstrate. So it is most, I think, important topic of the total session. So here some content also, this will be explained by the Anindita, and introduction also. So here some I will demonstrate some of the issues of unsupervised method and short-based learning method here. So uh, here only one thing I will describe, this one also 17, the zero shot learning process, and why it is called unsupervised. So I think interaction also most important here. So anyone tell me, uh, Devojyoti, Devojit, or our first year students, can you tell me the what is unsupervised method or learning method? Anyone? Okay, Jagrita, can you tell me what is unsupervised method? Jagrita,
Yes, sir.
acha, can you tell me about the what is unsupervised method?
So when the model is not previously trained and we just give the model basic things, and when it is not pre-trained, that time we can call it unsupervised.
Okay, so the model has been never seen this kind of data. Okay, so um, here the zero shot learning is, I think, one of the very much important example is shown here, zero shot learning. So if you, the problem, what is the problem? And in our hand to learn the picture of the zebra, the test image. So this is the problem statement. How can you learn this zebra? But we have the three photos, not zebra. One is horse, one is tiger, and another is penguin. These all the scene classes. But I think zebra is not seen by the machine. So can you imagine the problem here? The Ushmita, can you imagine the problem?
Yes.
what is the problem of the zero shot? That is the
Sir, I have
what is
came
the value?
currently I have came right now. I have
Okay,
joined right now.
okay. No issue. Okay, so this is basically it is the test image zebra, but we have no information of zebra. But how can we learn this image without any previous image of the zebra? This is called some to some extent unsupervised learning. But in a uh, very few uh, that is place or occurrence, we are also using some known cases also that is in that is in the target, and this will be uh, explained by the Onindita in the next slides or the full presentation. So horse is not like that, the zebra, tiger is not like that, the zebra, and the penguin also is not like as a zebra. So machine or the model is mainly trained by the contour of the horse, that is like a zebra. Then tiger, the strips of the tiger, same like another kind of strip is in zebra, and the color combination, black and white. This kind of information is from the panda. So I think after getting all of the attribute features, like the contour horse-like contour, strip like the tiger, and the black and the white color from the panda, the whole thing is summarized for the detection here. So I think is not the detection classification. So then summarize all the information from the attribute features. A test image is generated by the machine or the model. So this kind of hints and attribute of the unseen classes accommodate and use it for the unknown class to represent the unknown class. This is called as a unsupervised learning method. But in some instance or in some portion, we will also using some ground truth imaging, but percentage is very low. So now it may be a very good presentation. Uh, I am very much happy Undito will present it, and please see the programming knowledge also, and uh, how to write from algorithm to programming and hands-on. So it will be a benefit. It will be a very benefited talk, I think, for all of you. So Nindita, I will request you, please start.
Okay, sir.
Thank you.
Hey, Jagata, screen time share to stop.
Okay, now you can start.
Thank you, sir. Good afternoon to all. My name is Onindita Mohanto, PhD Research Scholar in Computer Science and Engineering Department, Tripura University. Today I will give a hands-on demonstration on short-base learning technique for handling unbalanced dataset in cancer stage diagnosis using microscopic cell imaging. Here is the list of content I will cover. First, I will give the basic introduction, then different microscopic imaging modality for cancer abnormality diagnosis, challenges of microscopic imaging modality for automatic cancer diagnosis, different method for cancer abnormality detection, few shot and zero shot learning technique, and the types of the few shot and zero shot learning techniques. As we know, cancer is one of the leading cause of death worldwide. In the year 2020, around 10 million people are die only because of the cancer. Among all other microscopic imaging technique, histopathological image analysis is considered as a gold standard for cancer abnormality detection. For that, automatic computerized analysis of histopathological imaging can help doctor and pathologist in making more faster and accurate decision. In table 1, we show the different type of microscopic imaging used for automatic cancer abnormality diagnosis. The first one is pap smear imaging, which is simple and widely used for early cancer detection, especially for cervical cancer. The second one is the histopathological imaging, which is the most common and reliable method. It is also called as the gold standard for cancer abnormality diagnosis. And the third one is the fluorescence imaging, which help to identify cancer area clearly, but it needs special equipment for image capturing. And the last one is the field emission scanning electron microscopic imaging, which provide detailed 3D surface images, but it is limited to the surface area. It can't visualize internal structure. However, each of these imaging technique has its own advantage and disadvantages, and they are used for different type of cancer studies. Table 2 shows the main challenges in microscopic imaging for automatic computerized cancer abnormality detection. These challenges are mainly categorized as cell-level challenges and environment-based challenges. Cell-level challenges include blurring, heterogeneity, artifacts in microscopic imaging, cell overlapping, etc. This makes it hard for computer to segment and classify cancer cell correctly. The second challenge is the environment-based challenge, which include poor image quality, unbalanced dataset, limited training dataset, high computational cost, etc. These factors affect the accuracy of the automatic cancer detection. Overall, all these challenges make it difficult to achieve consistent and reliable result in cancer abnormality diagnosis. Most of the common cell-level challenge is the presence of artifacts in microscopic image, which is the unwanted change or distortion in tissue images that make image interpretation difficult. Artifacts can happen due to the several causes like axal blood, inflammation in cell structure and ear dying, etc. Because of artifacts, it becomes more difficult to correctly identify nuclei or cell area which affect the diagnosis accuracy. The second most common challenge is the defocus blur, which happens when the microscope or camera is not properly focused. It is caused by small alignment errors, movement during image capturing, etc. As a result, the image looks blurred or the soft, and it becomes hard to see the cell or nuclear edges from the microscopic image. The next challenge is the overlapping cell or clumping. It happens when many cells or nuclei are too close or merged together in cluster. This is usually caused by dense cell population or uneven tissue preparation. As a result, the nuclei boundaries are become unclear and make it harder to separate and classify individual cells. The next challenge is the heterogeneity. Heterogeneity means difference in the size, shape, structure of nuclei or cell in a same microscopic image. It happens due to the natural cell life cycle change and the different in cell type. Because of this, nuclear cell look inconsistent, making automatic detection and classification more difficult. Among all these challenges, limited training dataset in microscopic imaging is the biggest challenge. To overcome the limited training dataset problem in medical imaging, we use few-shot learning technique. In the next slide, we will discuss few-shot learning technique in more details. What is few-shot learning? Few-shot learning is a method to teach computer to recognize something with only a few number of example or no example. Normally, machine learning need a lot of data to learn, but in few-shot learning, we only use a few amount of data for model training. It also allows learning to classify one modality of data using information from another modality. Why we use few-shot learning in medical image processing? In medical field, we do not have access to sample of all possible diseases for deep learning model training. Traditional deep learning model require a huge amount of data set. Few-shot learning can able to learn new diseases with few level example. If few-shot learning help to reduce the need of large data set, manual data annotation, expert dependency, time consumption in medical domain. It also help to deal with the shortage of data. Few-shot learning can be categorized as zero-shot learning, one-shot learning, and end-shot learning. In zero-shot learning, we have no images of the target class. In one-shot learning, we have just one image of a target class. And in end-shot learning, we have n number of images per class of the target and could be 1 to 50. And this is ideal for medical imaging where level datasets are very limited. First, we will discuss zero-shot learning, then the end-shot learning techniques. What is zero-shot learning? In zero-shot learning, we teach a deep learning model to recognize something it has never seen before. In this case, the model has no example or no images of the target object, but it uses knowledge from the other related classes to identify it. Suppose in this example, we have lion and otter class images in our training set, but we need to classify tiger class in our target set. How zero-shot learning works? In zero-shot learning, we train the model using images that already have class level. These are called the source class image or the scene class image. And we also provide some information or attribute like color or shape feature of the unseen class image or the target class image. During testing, the model used this attribute to recognize new classes or the target classes that has never part of the training data. So basically, the machine learned from a scene dataset and used these descriptive attributes to identify unseen classes. Figure 4 shows an example of zero-shot learning method. In this example, we see that in our scene class or our source class, we have donkey, horse, tiger, panda, penguin images. But in our target, we have zebra class images. Our deep learning model has never seen a zebra images before. So we only give the descriptive attribute of zebra class to our model, like it look like horse, it has black and white strip, etc. Using this attribute information, uh, The model mapped the feature of the scene classes to the target class, and it thus able to recognize the zebra class using the attribute information. So there are two main types of attribute used in zero-shot learning. The first one is manually defined attribute. These are the high-level characteristic like shape, color, texture, feature of our target. These attributes are more accurate, but it needs human effort to labeling. The second one is the word vector attribute. These attributes are automatically generated from deep learning model from using the large text source like the Wikipedia. They require less human effort, but it can be very noisy. Common deep learning model use are the word to vector or the node to vector method. Based on the test data, zero-shot learning problem can be divided into two major categories. The first one is the conventional zero-shot learning technique, and the second one is the generative zero-shot learning technique. Conventional zero-shot learning model learn the relationship between the visual feature and the symmetric information such as attribute or word vector. It tries to project both seen and unseen classes into a common embedding space so that unseen class can be recognized based on their similarity to the seen one. The conventional zero-shot learning are the embedding-based method, autoencoder-based method, attention-based method, etc. And the second one is the generative zero-shot learning, like GIAN-based method, where we generate synthetic or the fake images to overcome zero-shot learning problem. Figure 6 show one example of the conventional zero-shot learning model called level embedding for image classification. In this model, instead of treating class level as separate category, this method represents each class level as a vector in a shared space. This model learns to match image feature of these level vectors so that it can predict unseen classes. Here is an example of the generative zero-shot learning method called the feature generating network. In this method, the model creates artificial features for unseen class that have no images. It uses a generative network to produce this feature from label image information. These generated features are then used to train a classifier. Thus, it helps to recognize new unseen classes without any real image. Here is another example of the generative zero-shot learning technique, which is called gradient matching generative networks. In this method, we match the gradient between the real image and the fake images to make them more realistic. The generated features are helped to train classifier to classify unseen classes. As we know, few-shot learning problem, we have 1 to n number of images are available of the target class. But domain adaptive few-shot learning problem, the target class are not only poorly represented but also come from a different domain. In figure 5, we show 4 type of classification problem in deep learning. The first one is the conventional deep learning problem where our source class and the target class belong to the same classes. Also, the source class domain and the target class domain are remain same. The second one is the domain adaptation based problem, where our source and the target belong to the same class, but the source domain and the target domain are different. The third one is the few-shot learning based problem. In few-shot learning based problem, we have same source and target domain, but the class level are different. The fourth one is the domain adaptive few-shot learning problem, where domain adaptation problem and few-shot learning problem are occur jointly. Where uh, we have source class and the source domain are different from the target class and the target domain. Here we show different type of few-shot learning method which are used to overcome the few-shot learning problem. The first one is the data augmentation based method, which is called like a generative adversarial network, where we generate large number of fake images to overcome few-shot learning problem. But as we know, microscopic images are very sensitive. That's why using data augmentation based method are not very suitable for medical microscopic imaging. The second technique is transfer learning based method, like transactive fine tuning technique, where we learn from a source class and fine tune this information for target class classification. But it requires a large amount of source dataset, which is also not possible in medical microscopic imaging. Third one is the meta learning based method, which is further categorized as metric learning and the gradient-based meta-learning technique. In meta-learning based method, we adjust the learning strategy according to the learning situation of each task. That's why it is also called as learning to learn process. It obtains model architecture and the model hyperparameter by selecting top scoring network for the next iteration. Meta-learning based methods are more suitable for microscopic imaging as compared to the other few-shot learning method to overcome the few-shot learning problem in the medical domain. Here is an example of one of the most popular matrix-based meta-learning technique called ProtoNet, where each class is represented by a prototype, which is the average of its example in the feature space. After that, new data points are classified based on the distance to this class prototype. In prototypical learning, we first randomly sample a task from the task distribution. For each class in the support set, we calculate the prototype using the support samples. Uh, for each class in the query set, we update the feature extractor weight to minimize the distance of the query point. And we iteratively repeat the same step until we find the optimum meta parameter. Here are some examples of prototype-like algorithm like matching network and the relation network. Second one is the example of gradient-based meta-learning called the model agnostic meta-learning technique or MEMOL. MEMOL learn model parameter that can quickly adapt to new tasks with few examples. It fine-tunes one small support set, then tests on the query set. It forces the network to learn features that transfer well across different tasks. Here is the algorithm of MEMOL method. We first start a random meta parameter initialization theta and repeat until we converge. Then sample a batch of task tree. For each task ti in the batch, we fine-tune theta on the support set for k step to obtain theta ik. In the outer loop, we update theta based on gradients for the query set loss at the theta ik. In MAML, we have two type of meta optimization. In inner loop, we optimize inner parameter, and the outer loop, we optimize the outer parameter. The inner loop and outer loop optimization equation are given here. Some other model mammal-like algorithms are the first-order mammal and the reptile, which are the further updated version of the mammal architecture, in which we reduce the computational complexity and the computational time of our model. To implement meta-learning, here is the for domain adaptive few shot learning problem. Here is the basic background details information. In few shot learning problem, we have a source class and a target class. We reduce the class gap. In our target class, we have only 1 to 20 number of sample images per class. For domain adaptation problem, we have a source domain and a target domain. We try to reduce the domain gap. We measure the domain shift by the cancer type, image magnification factor, and the staining color. In our problem setting, in our source class, we have 5 different classes of lung and colon cancer are given. In our target class, we have different classes of breast cancer having only 1 to 20 number of images per class. In our source domain, we have lung and colon cancer histopathological images with unknown magnification factor and staining information. And in our target domain, we have breast cancer histopathological images with 4x, 40x, and 400x magnification factor images. Here is an overview of meta-learning framework for domain adaptive few shot learning problem. Our framework is first divided as two main stages. The first one is the meta training stage and the second one is the meta testing stage. In meta training stage, we train our model using our source dataset or our scene dataset. And the meta testing stage, we train our model using our target dataset, which is very limited, like 1 to 20 number of images per class. First, in our source, we divide our source data into m number of taxes. In each tax, we have support set which is used to train our model, baseliner model, and the query set which is used for model validation. First, we have m number of baseliner. In our baseliner, here we use the basic CNN network as a baseliner. We use m number of baseliner. We optimize m number of baseliner using randomly assigned taxes. After optimize the baseliner using assigned tax, we calculate the average loss and update the meta parameter to optimization. We iteratively repeat the same step until we find a meta optimized hyperparameter. After that, we go for the meta testing stage, in which we update or fine tune the meta hyperparameter using our target dataset. After that, we calculate the classification accuracy using our test image, which is belong to our target data. Here in table 6, we show the experimental result of different few shot learning and domain adaptive few shot learning method for same domain few short learning problem setting on the breakage dataset. On the breakage dataset, we conduct the performance evaluation in 3 different shot level, that is 5 shot, 10 shot and 20 shot. 5 shot means we use 5 number of images per class, 10 shot means we use 10 number of images per class, and 20 shot means we use 20 number of images per class, and 5-year classification means we use 5 different classes for classification. In the experiment study, we saw that protonate perform well in different shot level. Like in protonate in 5 shot setting, we have 66.85% accuracy. In 10 shot problem setting, we have 77.40% accuracy, and in 20 shot, we have 89.83% accuracy. We conduct the same experiment in the different domain few shot learning problem setting on the breakage dataset. In different domain few shot learning problem setting, we saw that mammal perform well as compared to the other existing few shot learning technique. We also conduct the same experiment in different domain few shot learning problem setting on the breast cancer IDC grid dataset. Here also we show that mammal perform well as compared to the other existing technique in the different domain few shot learning problem. Figure 15 shows a graphical representation of the performance evolution result of different few shot learning and domain adaptive few shot learning method for same domain and different domain few shot learning problem. Figure 15a. From figure 15a, we show that protonate perform well in the same domain few shot learning problem setting, but in the different domain few shot learning problem setting, mammal-based algorithm, that is GNet mammal plus meta fine-tuning technique perform well. Here are my use references. Thank you.
So if any kind of question, please. Or no, Uttara, any kind of question? Sushmita?
Yes,
Or
sir.
any?
Hello, sir.
Hi, yes, yes, Doshmi.
Sir, I just want to ask that we all know what happens to nucleus when cancer happens. So, what happened to axons and dendrite, the
Now,
receiving and sending signals?
axon and dendron is a neuron part. Is a totally
Yes.
neurological part. Okay, so it is a good question. The axon and dendron is a part of neurological neuron. Okay, and cell is the another issue. If you for the signaling, if you send the signal from one cell to another cell, okay,
Yes, sir.
then you have to use or they will help you axon dendron. So
No,
totally
after cancer, what happens to them? I'm asking that when cancer, no, somebody is affected by cancer. So,
yes.
how is the axon or dendrite work properly in that time?
Uh, they are the only the if you send the wrong signal, they will get back to the wrong signal. Okay, they are only the signaling part or the information bearing part. If the wrong signal or the that is the information is wrong,
Yes, sir.
if the any cell is sending that the
Yeah.
cell is cancerous, okay, and the
Yes.
sending a this kind of information, so it will bear this kind of information. So there
So
is a no
signals will be wrong in that time.
clear,
Signals will be wrong.
clear signal is wrong because cancer is such kind of thing is if you see the if you see the previous presentation of the Niharika madam,
Yes, ma'am. Yes,
you
yes,
can
sir.
see cell is working into that is the two region. You know why the actual good cells are helping the cancer cell. Okay,
Yes,
another
yes.
reason the cancer cell is not a very good person. He is also converting them in a good cell to bad cell. This is the two
Yes.
concept. So whenever they are converted into the bad cell or the wrong cell, they will usually use the bad information or the wrong information in the everything. Okay, then this will be the spread also.
Okay.
So I think it is
Yes.
also I think um, uh, that is the related question. Very good. So any other question?
Thank you.
Any other question, please? Okay. So many thanks, Unindita, for assisting me and a very good presentation.
Thank you, sir.
And everything is very. The few shot is a very complex thing also. Okay, so a workshop may be conducted with a few shot learning, no issue. So this is a primary and the preliminary fundamental concept of the few shot learning. So thank you to all. We will again meet tomorrow at 10 o'clock. Okay. So
Okay, sir.
Pooja, please formally end this session. Thank you to all.
Thank you so much, Dr. Miyal Kantik sir and Nandita Mohanto for nice tutorial session on future planning. So, and thank you the participants for listening the whole tutorial session and asking such good question. I think Dabashmita should ask this kind of biological question to ma'am also. She can deliver a very biological way answer of it. So this was a machine learning session, and I hope you understand everything. So we will meet tomorrow at 10 a.m. for lecture 7, clinical preclinical aspects, therapies, traditional and novel cancer stem cells and targeted approaches personalized with medicine. So here we are ending our third day. We, thank you so much everyone.
So thank you to all.
Thank you, sir.